Okay, so now domain Archaea constitute domains of single cell prokaryotes. Again, so when you know about prokaryotes, so there's definitely no nucleus. So they are initially classified as bacteria, but because of the what we call these the gene sequencing of their ribosomal RNA, so now scientists decided to detach it from, take it out from the bacterial and put them into new domain called Archaea. Okay, so the first observed Archaea are extremophiles. So they are extremophile. What's an extremophile? They always live in the very harsh environment, such as very high temperature. Okay, like hot spring, or in the lake with high pH. Okay, or extremely low pH, very acidic condition, or in the conditions that no oxygen at all, or in the condition with a very, very high surrounding salt contents. Okay, but nowadays we do realize that they not only live in the extreme conditions, they can live in other regions of the world as well. Okay, so they have cell wall, but they don't have peptidoglycan cell wall. Are you clear? They don't have peptidoglycan. So that's why peptidoglycan cell wall is a specific features. It's a specific features for the bacterial. Archaea, no, they don't have peptidoglycan cell wall. Even though they have cell wall, the cell wall basically assembled from a surface layer protein known as S layer, surface layer protein layer, S layer cell wall. Are you clear? They have S layer cell wall rather than the peptidoglycan cell wall. Okay, so but this is again extra information for you guys. If you want to name, put a structure, we say that they have cell wall, but the cell wall lack of peptidoglycan. So they do have the circular DNA, but special here, highlight this point. Circular DNA, but with histone protein. Can you see that? So it means that not naked DNA. Are you clear, guys? Not naked DNA. Okay, so they do, because they are prokaryotic cells, they do have the plasmid, they can carry out bacterial conjugations. Okay, so if you look at the morphology, it means the shape of the bacteria, Archaea, so they are similar to the bacterial. But you look at the gene and the metabolic pathway, they are more closely related to those eukaryotes. It's something like basically the intermediates. Okay, so transcription in the Archaea most closely resemble the eukaryotics, and also RNA polymerase very close to the eukaryotes. And Archaea was split off as a third domains because of the differences in their ribosomal RNA compared to the bacterial. They are most closely related to the eukaryotic cells or eukaryote. So therefore, scientists decided, or taxonomists decided to take them out and place them into a new domain. Okay, so again, highlight guys here. This is very important. Okay, again, because they are prokaryotic cells, they don't have membrane-bound organelles. So it means that they don't have mitochondria. Okay, they don't have chloroplast, nucleus, they don't have chloroplast, they don't have, okay? So they do have the circular DNA, but does have, the circular DNA does have histone protein. So this is a characteristic for Archaea, circular DNA with histone protein associated with it. So small circular DNA molecule, plasmid, yes, quite usual. That means in this Archaea, they have the 70S ribosome, but the features are similar to those eukaryotic ribosome, not the bacterial ribosome. Cell wall always present, but does not contain peptidoglycan. Cell divided by binary fission, not mitosis. Again, they are single cell or small group of cells, but they are still unicellular. Okay, these are characteristic features. So the one that very, very specific, okay, circular DNA with histone protein, where you won't be able to get this in the bacterial as well as the eukaryotic cells. Okay, so what are the phylums, okay, under this Archaea? So we do have the thermophiles. Thermophile basically means that they live in a very, very hot environments. Okay, for example, these Pyrorubus fumari, so they live in the temperature with 122 degrees Celsius in the Yellowstone National Park's hot spring. Okay, so some of them are very special. Some of them, okay, in a temperature lower than, let's say at room temperature, they freeze. They cannot survive, they freeze. Okay, because they used to survive in a very, very hot environments. Okay, so we do have the halophile. Halophile, basically they live in the high salt concentration called halobacterium. Okay, then we have the methanogen. Methanogen is the one, a very, very special organism. They live in our intestine, the large intestine. So they produce a methane gas as the metabolic byproduct in the hypoxia condition. Okay, so they are common in the wetlands and also in the digestive tract of the animals such as ruminants and humans. Okay, so this methanobacteria is the one that contribute to the methane concentrations increase in the methane. Methane is a greenhouse gas. Methane is a greenhouse gas. Okay, so when you fart, okay, contain a methane, right? So this methane actually produced by those these are methanogens. Methanogens. So they are very extreme. They can live in the, because our large intestine, okay, without the oxygen, but they can still survive, okay, through this anaerobic or hypoxic condition means anaerobic conditions. Okay, so they are the one that contribute to the methane gas release. Okay, so we continue to the next ones. Sorry, stop recording.